Hi, welcome to lecture number 2 for the skeletal muscle system that goes along with Hull's Essentials of Human Anatomy and Physiology textbook. So in this lecture, we will be speaking about how skeletal muscles contract at the biochemical level. So first off, there's a lot going on here. So you need to know what events occur and in what order. And the end result is the sarcomere, which is the smallest contractile unit, will actually shorten. The Z discs will move closer to one another, and the overall sarcomere will get shorter. And when you take sarcomere lined up with sarcomere after sarcomere alongside those myofilaments, then at the macro level, the entire muscle will get shorter and it will pull on its attachments, which is typically a bone. This is all done by the thick filament myosin protein connecting or binding to the thin filament, the actin protein. And that they will slide past one another, kind of like an accordion, and that's what causes the shortening. Okay. So myosin, it's a strand of protein made of many individual subunits. So it's actually two twisted strands, and then there are what we call head. It's in a glob-like shape, so it's a globular head that's projected outward along the strands for each myosin molecule, but then many myosin molecules will twist and bind together to form one thick filament, so it would have multiple of these globular heads. The actin is also a globular protein arranged in twisted filaments, but it does not have the heads. Instead, it has these indentations in it, which are the binding sites, so like a complementary puzzle piece where the myosin heads will bind. Now, there are some other proteins involved here. One is called troponin, and the other is tropomyosin. And these are found along the surface of the actin molecule, and they sort of kind of cover up and protect that binding site on the actin. So the thin filament is actually made up of 3 proteins types, although there are several different protein molecules that make it up. And then the thick filament is just made up of 1 protein type, the myosin. Here's a picture. So each of these strands, you can see, get the laser pointer out here. So this is just one strand here, and here's the 2 myosin heads. But you have many strands together that make up the thick filament. So you think of this kind of like a bundle of pencils, just microscopic pencils with the erasers sticking out, and they're kind of bent over. And then this would be the actin, the white part in the middle, that's the actin. And then here would be the troponin and the tropomyosin. Sorry, here would be the troponin. Let me show you the tropomyosin a little bit more clearly. It's this outer, thinner protein. Okay, so this part is a little bit complex, so just try to memorize the order of the steps. This is Nobel Prize winning work that took many scientists many years to figure out how these muscles actually physically get shorter. So the model that we're working on is called the sliding filament model. So filament because they're these thin little protein sticks, they look like a filament, and the sliding because they slide past one another. So the myosin head will attach to a binding site on the actin filament and form a cross bridge. Once those bind, that causes a shape change in the myosin and it causes the head to bend, which pulls the actin filament further towards the center of the sarcomere. And this will happen repeatedly. So it almost looks like, if you were to see this, it almost looks like the myosin is kind of walking along the actin. The head will release and then attach to the next binding site. So that's where you get that walking action going on. And over time, even though it's just a tiny little amount, when you add up all the sarcomere shortening, you get a great distance of shortening. and the whole muscle shortens. This uses a lot of energy from the cell in the form of ATP. So in order to do muscle contraction, your cells have to take ATP and break it down into ADP and inorganic phosphorus, P sub I. Okay. ATP isn't free. Okay. ATP has to be remade constantly. So there's an enzyme called ATP synthase. It's abbreviated ATPase. This enzyme is found in the mitochondria, and it makes new ATP. Basically, it takes the byproducts of muscle contraction, the ADP and the inorganic phosphorus, and then recombines them to make ATP. This is one reason why skeletal muscle cells have so many mitochondria, because they need to make a lot of ATP. And you can't just store up ATP continually. It will break down after a little while. So it's constantly having to be remade. The ATP, what it does in the muscle contraction is actually it relaxes the muscle. So if there is no ATP available, the muscle will stay in the contracted state, and this is called a muscle cramp. You need the ATP to relax the muscle and reset it. This is also what causes rigor mortis. After someone has died, there's no more ATP. Like death is cell death, no more ATP is being made. So the muscles just stay contracted. Make new ATP if you're still alive and you have a muscle cramp. Yeah, usually the problem is you have an oxygen debt. You've exhausted all of your supplies of oxygen, so you need to rest and breathe in deeply and then work out that muscle cramp. So here's the sliding filament model. It's a cycle. So the first step, okay, I'm going to start here when the muscle contracts, is the muscle fiber will have received a stimulus from a nerve, and that stimulus caused calcium ions to be released. This is a calcium ion. It has a +2 charge. The calcium ions are released, and then they bind to troponin, which causes the tropomyosin to be pulled aside as well, and the binding sites on the actin are then exposed, and then that attracts the myosin heads. So the myosin heads will attach to the actin binding sites and form what we call a cross bridge. Then there were already ADP and P attached here, like at the beginning. Then the ADP and P are released. And the myosin and the cross bridges pull the actin filament towards the center. This is called the power stroke. At that point, the ADP and P go off back to the mitochondria to get replenished, and a new ATP molecule is needed, and it binds to the myosin, which causes a shape change of this myosin head and causes it to detach from the actin filament. And with these heads detached, the actin filament can just snap back into its original configuration where it's relaxed. At that point, the ATP splits into ADP and an inorganic phosphorus. So the cross bridges are cocked. We say they're ready for the next cycle. Okay, and then it would just repeat over and over again. So here's what it looks like. If you were to look at the sarcomere here, this is a kind of a stylized cartoon drawing here. The relaxed is at the top and the contracted is at the bottom. So the myosin itself doesn't move, that stays put. It's the actin that gets pulled in. So if you can see in the relaxed state, the Z lines are here. But if I just draw a straight line down, you can see the Z line was here, but now everything's been kind of moved over to the left or to the right, depending on where you are. You can see there's now overlap. Okay. You can also see this. This is a slide of a muscle tissue. You could see this happening in real time under the microscope. So you could actually see the Z line here. It started here, but you can see it's moving further and further towards Now, so that's within the muscle fiber itself, okay? So what causes the calcium to be released? So we have to take a step back and we have to go back to the motor unit where a neuron, where the axonal ending of a neuron actually makes a synapse with the muscle, okay, at the motor end plate. Inside that neuron, neurons communicate with one another with many different neurotransmitters, but they communicate specifically with muscles using the neurotransmitter named acetylcholine. So you do need to know the name of this neurotransmitter, acetylcholine. Sometimes we abbreviate it capital A, capital C, little h. So the motor neuron is actually responsible for making this chemical. It gets stored in the axon in vesicles. We call them synaptic vesicles because they're very nearby the synapse. So the acetylcholine is stored there and it's just waiting for the neuron to tell it to go and do its job. So if the neuron receives the message from the central nervous system that this muscle needs to contract, the neuron will release that vesicle. It will send the synaptic vesicle to its plasma membrane and it's going to go through the process of exocytosis and release acetylcholine outside of that neuron. The neuron is done with its job at that point, and the acetylcholine actually has to physically drift across the synaptic cleft. It has to diffuse until it reaches the plasma membrane of the muscle fiber, and that plasma membrane has a special name. It's called the sarcolemma. There are special protein receptors on the sarcolemma that respond to acetylcholine. So when the acetylcholine hits that protein receptor, it's going to trigger an impulse within that muscle. It's going to start an action potential that moves down the sarcolemma into the sarcoplasmic reticulum, and that's going to cause the SR, the sarcoplasmic reticulum, to release calcium ions that it had stored up there. It'll release those calcium ions into the cytosol. The higher, now suddenly higher concentration of calcium ions is going to interact with the troponin and the tropomyosin and move those aside to expose the myosin binding sites like we talked about on a previous slide. That causes the cross bridges to form between the myosin heads and the actin binding sites. The actin gets pulled, ATP gets used up, and then the whole sarcomere shortens. This is going to continue. This is called a muscle contraction. It will continue for as long as the nerve impulse tells it to. So if, if you're carrying a heavy box, that's going to continue until you decide to set the box down, or it's a really heavy box and you're trying to see how long you can carry it. Eventually, the muscle will actually become fatigued and spent and you'll drop the box involuntarily. But typically, we take some action before that happens. Once the nerve impulse stops, then the muscle can relax, but you have to get rid of the acetylcholine because the acetylcholine is still kind of drifting around in the synaptic cleft, kind of playing bumper cars, and it can repeatedly hit the receptor proteins on the sarcolemma. So the acetylcholine has to be cleaned up. And how that happens is an enzyme called acetylcholinesterase. So enzymes always end in ASE, and a lot of times they're named after what molecule they act on. So the acetylcholinesterase breaks down acetylcholine so that it's no longer active and the muscle fiber is no longer being stimulated. It decomposes, basically. Sometimes we abbreviate that enzyme ACH, ACE. So A, capital C, little h, little a, little s, little e. Once that happens, over time, the calcium is going to be returned to the SR, the sarcoplasmic reticulum. It takes ATP to do this. You have to reset those muscles. And then the ATP will also bind to the myosin heads, and that will break the link between the actin and the myosin. And the actin is going to slide back to its original position, and the muscle is going to relax. So here are the steps. written down in order so that you could review yourself. And then here are also the steps of muscle relaxation. These are all found in your book. You can use them to study with and to work on your practice work with. And that is the end of lecture 2.